Hello, and welcome to our next webinar in our series. Today we have three speakers speaking on the topic of the plant cell atlas. And the first one is Hilda, the second one is Daniel, and the third one is Jen. And we are hosted and moderated today by Kenneth. This webinar is brought together to celebrate the February 2022 Plant Physiology focus issue on the topic of the Plant Cell Atlas. And this focus issue is edited by Kenneth Birnbaum, Julia Bailey-Saras, Marissa Utegi, and Sue Ree, and it's currently online. You can find it at the Plant Viz website. You can find this and many other recordings of webinars will be available on YouTube shortly on the Plante YouTube channel. We'd like to make a special thank you to ASPB members. If you're not a member of ASPB, we encourage you to join. The membership supports this series. We have a code presents10 that will give you 10% off your membership dues. Please put questions for the speakers into the questions box, and they will be read out by Kenneth after each speaker speaks. If you have technical questions, feel free to email me, mwilliams@aspb.org. And now I'm going to turn the microphone and the camera over to Kenneth. Thank you.
Okay, great. Thank you, Mary. And thank you all for coming. I first just wanted to explain what is the Plant Cell Atlas. I think that would be a good thing. And technically, we define this as the quest, the mission to identify all plant cell types and characterize their function. So this is a big task. And clearly sort of implied in this, there's a technical side that we are looking for innovations or adaptations to techniques that are going to address specific biological questions in the plant kingdom. So very much geared toward the plant kingdom. And you will recognize by the name that it's sort of fashioned after the human cell atlas, but in some senses, we want to go beyond that, as you'll see today. And of course, these are very much geared toward plant questions. Okay, so the focus issue that Mary was talking about had 17 articles and reviews, and we encourage you to go look at that, as Mary did. But today, we can't give you all 17, so we're giving you, let's call it a tasting of some of the things that we think will inspire you, and hopefully interest you, and sort of are their little treats out of that. And so let me stop talking and turn it over to the people who are going to talk about the science. And so the first speaker up today is Hilde Nelson. She's a group leader at the VIB in Ghent in the plant science biology department. She's interested in growth, and she uses the maize leaf as a model, as you'll see. And she's going to tell us about spatial transcriptomics and the plasticron 1. And I will turn it over now to Hilde. Go ahead.
Okay, thank you, Mary and Ken. Good morning, everybody, and good afternoon if you are in Europe or in these parts of the world. I'm very grateful with this opportunity to highlight the paper that we had in this special issue in Breakthrough Technologies, which was mainly focused on the in situ sequencing approach, which is only one of the ways to do spatial transcriptomics. I've altered the title of this presentation a little bit because I want to go even beyond what we published in this one paper to show you what the possibilities are with spatial transcriptomics. So as Ken already indicated, we're very much interested how a plant grows, and we use maize as a model system. So typically a lot of the traits that are measured are endpoint traits. And so for instance, final plant height. But if you measure this, it actually has a lot of growth processes that were occurring in order to achieve this final plant height. And this growth occurs in the individual organs, but also we've shown, and others also, that there is a high coordination between the growth processes in the different organs, for instance, between leaf, stem, and inflorescences. And so we've mainly chosen the maize leaf to study these growth processes. because it is quite easy accessible, it has a rapid turnover time, and also because it has a nice spatial organization, because the dividing cells are at the base of the leaf, followed by the elongating cells and then the mature cells. And so this allows us, because of the size of the maize leaf, to really sample for cells that are enriched for these dividing cells or elongating cells. And as such, we can do a lot of molecular downstream analysis. And so we combine these macroscopic phenotypes with the cellular analysis and molecular analysis to further understand how these growth processes are coordinated. And one of our pet genes that we have identified as such is called plastognon 1 or PLA1 in short, and it's part of a family of cytochrome P450s, which are known also in other plant species to stimulate organ growth. And in maize, we identified this when we tried to overexpress plastognon 1, we found that the plants were producing leaves which were much broader and they kept on growing. These plants had difficulties to make inflorescences, so we had to go back and make another design. And then we chose to express plastognon 1 ectopically within this division zone of the maize leaf, using a specific promoter that we identified through our profiling studies. And when we did this targeted overexpression, we actually saw that we could indeed stimulate organ growth in a way which was beneficial, like it's shown here for seedling vigor, but also later on when we tested these plants in field trials, we could show that it had a positive effect on other organs, and so it affected final biomass and seed yield. So this really showed to us that plastognon 1 had a lot of potential, so we further wanted to understand how its mode of action is. And so when we looked at the cellular analysis, we saw that plastognon 1 had a mode of action through making cells dividing for a longer period of time. And so we were thinking maybe it keeps the cells in an undifferentiated state for a longer period of time, but we couldn't really understand how it was doing so. On top of that, there were some other clues that we got that also pointed for us towards a role in the shoot apical meristem, besides the possibility that it regulates undifferentiated cells. Plastognon means that it regulates actually the time between leaves to be formed. And as you can see, in this case, this is a knockout mutant of plastognon 1. You see that shorter leaves are being formed, but that more leaves are being formed at the same amount of time as the segregating wild type. And this becomes progressively worse as plants develop. And as leaf initiation is also a process that takes place in the shoot apical meristem, we had another hint that the plant might function in the shoot apical meristem. And finally, there was a third hint that led us to the shoot apical meristem, and that was when we profiled the expression of plastognon 1 over this leaf developmental gradient that I explained. And then we found that plastognon 1 was very highly expressed in the dividing cells, but that it dropped almost to 0, even in cells that could still identify as dividing. So this means that plastognon discriminates between dividing cells close to the shoot apical meristem and more distant. And so for this reason, we really thought that we should check out the shoot apical meristem. And we did so by using in situ hybridization with plastognon 1 as a probe. And there, in this transverse section, we found that plastognon 1 was not expressed in the emerging primordia, in the P0, which you can see from this horseshoe-shaped expression profile. And we could confirm this in transverse sections, so the absence of PLA in the emerging primordia. However, we see that PLA is specifically expressed in the cells that subtend the emerging primordia. And when we zoom out a bit, we can see that this expression goes down as the primordia become older. And so to us, it looked more like plastognon 1 was a spiral around the shoot apex. And so this was very intriguing to us. And we wanted to know how the expression of plastognon 1 would correlate to known marker genes. Very much at that time, we got the opportunity to actually test in situ sequencing technology, which is a spatial transcriptomics technology that allows to visualize the expression of multiple genes simultaneously on the same tissue context. And based on, for instance, the laser capture microdissection data of the Scanlon lab, we picked a panel of 90 genes with high and low expression levels. Based upon literature, we took genes with very specific or broad expression levels, and we looked whether we could apply this technology also in plants, because until then it was mainly used in animal tissue. And as you can see from the image, this really worked well. For the majority of the genes, we could not find any correlation between the genes that did not function and the level of expression or the specificity. And so when we did this and we brought this technology to plants, we implemented another improvement, and that was in order to map the expression level to the individual cells. So what you see here are general expression profiles, but when you can segment different cells in the background, you can actually map all these count to the individual cells. And in animal tissue, this segmentation is typically done by DAPI staining to visualize the nuclei, and then it's thought that the cell is a certain radius around the nucleus. However, in plants, we know that the nucleus is not always nicely in the center of the cell, and so we complemented the DAPI staining with the cell wall stain. So once we had evaluated the technology in plants, we could use it to do some biology, and then I circle back to our favorite gene, plastochron-1. And so here I only showed 3 of the 90 genes because it becomes a little bit crowded sometimes. And what I want to show you here is that the expression of plastochron-1, which is in yellow, which is again the spiral down the shoot apical meristem or down the shoot apex, it actually coincides with a boundary that is being formed by the expression of the red gene and the blue gene. And the red gene is actually rough sheet 1, which is an ortholog or a homolog of knotted 1, which is a marker for indeterminate cells. And the blue marker is actually a marker for the epidermis, which means that really a cell fate has been acquired, and these are determinate cells. And so you can see that there is a sharp demarcation between the red and the blue gene, and that actually plastochron-1 is expressed at the boundary between these indeterminate and determinate cells. And so this fit actually with the phenotype that we saw in our targeted overexpression line, where we could show that indeed we saw a longer and higher expression in older leaf primordia of plastochron-1. It also ties in with what is known in literature, because in the single cell data sets that were published over the past couple of years, plastochron always turned out to be in the same cell clusters as, for instance, knotted 1. And so we are now further investigating the link between knotted 1 and plastochron-1, because also about 10 years ago, PLA1 was identified as one of the putative knotted 1 targets. And so this has led for us to a working hypothesis that we are further following up on. So
Hilde?
once we had,
Sorry,
yes,
3 minutes.
Yes, once we had now the system optimized for in situ sequencing, we also saw whether we could apply this beyond the paper that we published and for other technologies of spatial transcriptomics. And here I show that we could also make it work for molecular cartography, which typically has a smaller region of interest that can be imaged with a higher resolution. When we complemented this with the segmentation, we were able to really map the expression counts to the single cells. And as such, we can do a clustering and a co-expression analysis. And this analysis showed us that plastochron-1 was co-expressed with angustifolia 3 or GRF interacting factor 1. This has been shown in the past in Arabidopsis to be a growth regulator, but also by the team of Sarah Hake, it was shown that GIF1 plays an important role in meristem determinancy. And therefore, we actually decided to make a double mutant between plastochron-1 and AN3 or GIF1. And as you can see in the double mutant, we actually have a more severe phenotype than in the individual mutants. and especially when you pay attention to the amount of leaves that are being formed. Then again, we went back to the spatial transcriptomics to further investigate this genetic interaction. And so we used spatial transcriptomics with the same gene panel that I showed earlier in wild type and compared it to the mutant background. This is one flavor I can give you where you can see that the expression of plastochron 1 is altered and is more expanded into the dome of the shoot-buccal meristem in this GIF1 or angustifolia mutants. And we could actually confirm this again with classical or single gene in situ hybridizations. So with this, I hope I have convinced you that spatial transcriptomics is a very nice tool that we now can, a targeted approach that we can use in developmental biology, either to compare expression profile between different genes. We can do a gene targeted approach. We can look in which cell types is our gene expressed, but you can also do a cell targeted approach where you click on certain genes and you know which genes are defining this type of cells. However, when you map it to a single cell level, you can even go to higher order co-expression levels. And I think it goes without saying that you can use this technology to, for instance, annotate single cell clusters that you obtained. And so with this, I would like to thank the team that did this, and especially also all the collaborators, especially the people of the platforms that I mentioned and the funding agencies. And then the people in the team that did the work was pioneered by Reinout, who graduated 2 months ago and is now taken over by Jessica and Dania with the assistance of Julie and Kirin. And with this, I would like to take questions and hope I stayed in time.
That was great, Hildine. You're right on time, so thank you. Okay, so we have a few questions piling in the Q&A. If you put them in the chat, put them back in the Q&A and I will try to get to them. And so quickly, we have a couple of questions on resolution, and I guess we'll compare the two techniques in situ sequencing and the other spatial transcriptomics. And I guess we're probably talking about subcellular level.
Yeah, these providers don't the subcellular level. This we didn't really look at. I think we found it already a challenge to map it to individual cells, but I think it is possible to go to subcellular level. Yeah.
And Hilde, you mentioned, sorry.
Yeah, we didn't do it yet.
Okay, but you mentioned that the second technique had higher resolution than the in situ sequencing.
Yeah, that is correct. In our hands, it had a higher resolution, which means that we had per cell more counts at the gene level, which then allows to do much more, it gives you much more statistical power to do this co-expression analysis. However, the in situ sequencing has the advantage that the region that you can image is larger. So for some applications, you don't always need to go to a single cell level. So it depends, I think, which provider to use, which question you're trying to answer.
Okay, good. We have another question about PLA1 and whether it's doing the same thing in roots.
Well, to our knowledge, plastochron 1 is not expressed very highly in roots. Also, and this I definitely know, the counterpart in Arabidopsis is not expressed in the roots. However, we do see root phenotypes in our overexpression lines, especially also in Arabidopsis. When you use a 35S overexpression line, you definitely see phenotypes in the roots, but that's of course because you ectopically express it there. Normally, it's not expressed in the roots.
Okay, and maybe one last question here on PLA1, which is, do you have a hypothesis on how the localized expression of PLA1 can affect leaf growth so much further from the shoot apical meristem? Is it a mobile signal?
Yes, so that is indeed one of the big hypotheses that we have, because plastogron 1 is a cytochrome P450, which means it's an enzyme, it hydroxylizes something. Unfortunately, or maybe fortunately, because that gives us still something to work on, we don't know exactly what the molecule is that is being catalyzed by plastogron 1 and the orthologs. And so, but definitely it has been shown in other species that it is a mobile signal. It's thought to be, maybe a type of hormone or, so definitely that's one of the ideas. On the other hand, as you see that it is on the boundary between cells, you could also see it like a shuttling, so that it starts to initiate another signal cascade downstream into the leaves.
Right. Yeah, interesting. Okay, cool. All right, good. I think we should move on and stay on time. Thank you very much, Hilde. That was great, interesting. And while we're getting the slides up, let me introduce our next speaker. It's Daniel Kierzkowski. He's an assistant professor up in Montreal, and his work in general focuses on molecular, cellular, and tissue level mechanisms, really looking at plant shape, plant organ shape, and biomechanics. And he's going to talk to us about the, he's going to talk to us about more about, now we're going on the imaging side and tracking imaging of the growing stamen and the dynamics. And I don't see the slides up. Are we having an issue?
Yeah, it's coming.
Okay.
So thank you very much, Ken, for the nice introduction. Thanks for inviting me here, and thanks to everybody for being here with us. So today I would like to show you how live imaging can help us to understand growth and development of organs, specifically floral organs that are hidden inside the floral buds. One of the most fascinating questions in developmental biology is to understand how a group of undifferentiated cells can give rise to this tremendous variability of organ shapes that we see in nature. Organ forms emerge from intertwined interaction between different developmental processes. Cells has to grow in order to make the bigger organs. Organ has to be patterned, so different parts of the organ has to get some specific identity, so the cells has to differentiate as well. Those processes are controlled at cellular level via molecular molecules like hormones, genes, but also by physical forces, especially in plant tissues where cells are interconnected by stiff cell walls. If you look at the organ, for example, this Arabidopsis leaf, it's very difficult to infer how this organ has developed. What are the growth patterns leading to the emergence of this specific shape? One of the reasons for this is, for example, is that different growth patterns can actually generate the same forms, as exemplified in this simulation. So we really need to go and observe cellular behavior over time to understand development of organs in plants. So how do we do that actually in our lab? So we use confocal time-lapse imaging combined with morphographic software, which is an image analysis software developed by Richard Smith's group. And we acquire sequential images of the living tissue. In this example, it is a shoot apical meristem tomato with some time intervals. Using cell outlines, for example, membrane marker or cell wall stain, we can now segment those images on the surface into cells. And this helps us to generate heat maps of different growth parameters. So what you can see here, for example, is a heat map of growth, other way, area extension, where you can see that cells that are growing very quickly are marked in red, and cells that are growing slowly are marked in blue. So, for example, future emerging primordium, here, what I show you, is growing very quickly. So we have all quantitative information for all cell behaviors of entire organ during its development. And such an approach has been extensively used to monitor leaf growth. And recently has been also used to look at the growth of floral organs, more specifically at sepals. So sepals are very easy to access, and people has been looking at the growth of those organs in the past. But we are interested in more challenging, more difficult organs that are more difficult to access. So we want to look at the development of stamens, so male reproductive organs of flowers. So in Arabidopsis, you have 6 stamens, 4 longer stamens and 2 shorter stamens. And they are basically composed of 2 distinct regions. At the bottom, you have a stalk that is a filament. This filament will be elongating a lot to deliver pollen to the stigma. And at the top of this filament, you have anther, which where actually the pollen grains are developing. So if you do a cross-section for this anther, we will have 4 lobes. In each lobe will contain locule where the germlines will develop. Until now, stamen development has been basically inferred from fixed tissues. So basically, people were using histological sections to understand how those cells are working together in order to create the forms that we see. And the reason for this is basically that entire development of the stamen is hidden within the inside the floral bud. So we don't have easy access for confocal imaging. So how can we look at the stamen development with confocal imaging if this is hidden? So the postdoc Silvia Silvera in my lab has developed a new approach to do so. So she's dissecting inflorescence of Arabidopsis. She's getting into the flower the floral bud when we know that the stamens are initiating. She's very gently removing abaxial sepal, uncovering the initiating stamen primordia, and now we are able to follow this the growth of those organs with confocal microscope. So here what you can see is that we can now follow the development of this organ from its very early initiation, basically a very tiny primordium, until the final shape of the stamen is fully developed. So the same what you have seen in the movie is decapitated on this image. So we can see we are actually observing the same stamen from 1 day after initiation, tiny primordium, 30 microns, until 10 days after initiation, when you have a fully developed organ with different parts, and it's nearly ready to deliver pollen. So it's more than it's 1.5 weeks that of the imaging that we can now perform. So what can we learn from that? Quite a lot of things. Of course, I don't have time to go into details in this talk, but I will give you some examples. So for example, we can try to understand how many cells are giving rise to the primordium, to the stamen in the epidermis. So what you can see here is this floral meristem, where before any signs of stamen primordium initiation, and 1 day later, we have a clear bulge indicated by the positive Gaussian curvature. So if you now mark all the cells at this very early primordium and trace them back to the floral meristem, we can say, for example, that, okay, around 28 cells in the epidermis will be contributing to this organ, which is the number very similar to leaves or sepals. We can also now precisely determine the timing when different parts of the organ are specified. So filament, when actually the filament and anther are getting distinct fates. To do so, we can use the cell clones. Basically, we mark the cells with specific color at the very early stages, so at 4 days after initiation in this case, and we look how they develop over time, and we look at their sector, so their progeny. And so if we look at this progeny at the last stage, we can see, for example, at this specific time lapse, that all the sectors that developed from single cells at the very early stages will be either located at the filament or on the anther, indicating that this is exactly the stage when the 2 fates are specified, the filament and anther. So we can see it here colored in blue for anther and in yellow for filament. And this is very clear now, for example, that at this very early stage, when the primordial is still very tiny and looking like a finger, we already have those fates specified, and the filament that will be very, very long structure, up to a few millimeters, is actually developing only from 2 to 3 rows of cells. So how this filament elongation occurs, we can now look in details into that. And so, for example, what we decided to do is to see which part of the very early filament is contributing the most to the final length of this structure. So we marked the early filament, we divided into 5 equal parts, 20%, and we have looked at it, what happens over time with them. And strikingly, the 2 top parts, so green and yellow sector, that represented about 40% of the initial length, they don't grow. This part is not growing. So this part is not contributing to the filament. In contrast, the bottom parts, like the red one, this is the part where the growth actually occurs. So we can look at this even in more detail. So what you can see here is the heat map of growth for every single cells during the entire elongation of the filament. So again, just to remind you, red cells are growing fast, yellow are growing slowly. And we can see that at early stages with very fast growth, which is slowing down progressively to increase again just before anthesis. If we look at the cell proliferation, so cell divisions, we can see that they occur at early stages and they progressively are eliminated. So the last stage of growth, there's no divisions at all anymore. So we can actually, looking by that sort of data, we can distinguish 3 phases of growth of the filament. At the first phase, cells are growing very quickly, cells are dividing a lot, and all the structure is elongating. Later on, in the middle phase, the cell divisions are actually restricted to the bottom. The growth is also restricted to the proximal part of the filament, and the top part is not growing. So we have sort of a restriction of growth to the proximal part of the structure. Later on, we have acceleration of growth. So this last stage is there's no divisions. We have very, very fast growth, but this growth is not anymore located at the bottom. It's more displaced toward the tip of the filament. So it's like it's a very similar behavior as we can as it has been observed in the hypocotyl of Arabidopsis or Arabidopsis, for example. So we have the 3 different stages of the elongation of the filament. So what I have shown you here very quickly is that our approach enables really precise tracking of cells to understand the origin of different parts of an organ, or organ itself. And this enables us to also very precisely characterize the growth and development of the organ at cellular resolution in the quantitative ways, to basically put numbers to every single cell and understand how they interact with each other to generate a specific form. So this research actually provided the first quantitative atlas of cellular growth dynamics of any kind of the internal reproductive organs in plants from the initiation until their full development. So this is the first quantitative atlas of cellular growth for Stamen in Arabidopsis. So this work was done mainly by Silvia Silveira, a postdoc in my group, who has designed the approach and done all imaging, all analysis. And so Silvia was helped by Constance and Andrea in this work. This work was done in collaboration with Annie's Routier lab from University of Montreal. And of course, it was possible thanks to the funding from different Canadian funding agencies and from Quebec. So I would like to thank you very much for your attention, and I will be very happy to get any questions you would have. Thank you very much again.
Okay, great. Thank you, Dan. I didn't even give you your 3-minute warning because you were so tidy with your talk. You were on conclusions. Anyway, all right, let me start with some questions here. And let's start with this one, which is very intriguing. Can we also examine fertilization and reproductive processes using live imaging like this?
So I guess that fertilization, yes, you probably can do it, but this is not the same approach, quantitative approach. I mean, the pollen tube imaging is done already, yeah. But what I think what we will be able to do in the future is to, for example, look at the development of the internal tissues. So, for example, the germlines. Like we hope that one day we'll be able to go inside the developing anther and to understand how those different layers, endothelsium, etc., are forming to generate those the germlines.
Okay. And another question here, thank you. Does the microdissection that I guess you have to do on the outer organs change the physics component to which you referred early in the talk, I guess?
So you of course, this is, you cannot call this technique non-invasive. Yeah, we cut the tip of the plant, we dissect it, and so on. So for sure, you are expected to have some alterations, and we can in some time-lapse imaging that we do, we can see it. Yeah, for example, the locules that are very elongated will be less elongated in the in vitro cultures than on the plant. But in general, the growth, all the parts and components of the organs, especially if such a fertile organ like stamens, are in place. So we believe that it is a good extrapolation, and we are not actually affecting so much the growth or the shape of the structure in vitro cultures by removing other floral organs.
Okay, we have a lot of questions here. Let me just ask one of the first, and we have some time because you ended early. How do you, maybe you want to go into a little more detail of how you measure the growth speed in your metrics that you show with the heat maps? Okay,
so you have consecutive images. Those are series that are taken every 24 hours. We segment cells at the specific cell, let's say, at the time 0, and then we are looking at the progeny of those cells at time 1. Our morphographic software can give you basically the surface area of the cell at the beginning and their progeny at the end. And then in this way, you can calculate the area extension in percent. You can calculate the growth anisotropy. You can calculate how many cells originate from this one single cell, etc. And you can do it also for a very prolonged time. You can calculate how many cells do you get from one single cell at T0, and then how many cells you get it from it 5 days later. Yeah.
Okay. And then here's a question, and I was also wondering, you do this tracing back to the origin of when a cell gets its identity, but of course it would be nice to see the factors, the induction, the hormones, whatever, that are perhaps responsible. So the question here is, have you tried looking at the proteins, I guess, fluorescent or otherwise, that are also, in play during this growth process?
Of course, for this specific research that is already published, we have concentrated our efforts fully on quantifying the growth and proliferation. But yes, we are looking at this as well. We are interested in especially in auxin signaling and how the auxin would be, correlating with growth in organs, not only stamens, but also in other organs, because especially auxin is, on one side, is a hormone that is involved in patterning of an organ, but at the same time, it's also involved in the differentiation of different tissues, so accelerating or decelerating differentiation. So yes, it is on our table, and we will be doing this in the future, and we are doing it already.
Okay, good. And you just answered a couple of other questions in the answer to that. So you're very efficient with the questions. Let me move on to our last. Well, let me first thank you very much, Dan. That was a great talk, really interesting. And a lot of the comments also reflect that. And now I'll get to
Thank you very much.
Thank you. I'll get to our last speaker, Jen Brophy. She's an assistant professor at Stanford, and she's also a Chan Zuckerberg BioHub investigator. She's been interested in synthetic biology for a while, developing tools for engineering undomesticated bacteria and soil microbes. In plants, she did spatial pattern engineering with synthetic circuits. And now in her own lab, she's interested in applying synthetic biology to both plants and bacteria, and in particular interested in issues of climate change. And so Jen seems like she's ready to go, so I'll turn it over to her.
Okay, thank you, Ken, and hello, everybody. I'm going to tell you about what I wrote in a review for this Plant Cell Atlas issue. It is a perspective piece presenting an idea of how we may be able to engineer some synthetic plant development. I just want to make it really clear at the beginning here that this is an idea of how we may go about engineering plant form, and that it might not work, but I thought it would be useful to write about so that we have the opportunity to discuss this potential approach. Okay, so I want to start by talking about why we might want to engineer plant form. I think a lot of us are familiar with this idea that a lot of the foods that we eat today are derivatives of wild varieties in which we have changed the structure of the plant to increase both the size and some of the features of the edible portions of plants. So if we can continue to engineer plant form, we may be able to do things like continue to boost yield. But I think it's fun to consider other methods or other applications of engineering plant form. For example, waste reduction. Can we take some of the forms that plants grow into and reduce them down into shapes that are all suited for market, so that we don't end up wasting any of the produce that we grow? Or can we change the growth programs so that the plants that we produce are ideal for mechanized harvest? So I'm showing here saffron flowers, which are one of the most expensive food crops we have today, in part because it requires hand harvest of all of the portions of the plant that we like to eat. And so if we can change the structure of this flower so that it presents the portions that we want to collect in a way that make them amenable to mechanized harvest, we may be able to reduce the cost and labor that go into producing some of the food crops that we rely on. And then finally, maybe most dear to my heart, is stress tolerance. If we can change the structure of the plant, we may be able to produce varieties that are robust to environmental stress. So this is an example in which a root system may be a little bit too short to reach water during droughts where the upper layers of soil have worn out or dried out. And if we can engineer the root structure to reach water that's deeper in the soil, we may have plants that are more robust to something like drought stress. Okay, so this is the why of engineer plant form. The question is how. So I'm going to present an idea of how to engineer plant form, but I just want to highlight some really recent and interesting methods for engineering plant form that have been really impactful. So first, there's been a number of papers, both out of Cold Spring Harbor in Zach Lipman's lab and Dave Jackson's lab to engineer the promoter regions of important genes to change the structure of tomato and corn. There's also been efforts to express developmental genes in really specific spatial patterns so that you can alter the structure of the resulting plants, and methods for kind of dynamically changing gene expression by delivering viruses that encode Cas9 activators and repressors. And those Cas9 activators and repressors up or down regulate specific genes as the plant is growing and modify its structure. Now, this is all great, but we know that structure is encoded by a really complex network of genes. And so it becomes difficult to imagine how you might go about changing the structure, especially because as you modify expression of one gene, you may affect several different aspects of development. So I want to introduce a concept of refactoring to tell you how I think we can do this. So refactoring is a term borrowed from computer science, where you change the underlying code for a specific process such that the process remains the same, but the code is different. And I think that this is interesting because in biology, in synthetic biology specifically, we've applied this concept of refactoring to get control over complex biological systems. So in biology, refactoring means taking all of the genes that encode for a specific function and recoding the DNA sequence for each of the proteins. So you can use the degeneracy in the genetic code. So there are multiple codons that encode for each amino acid or most of the amino acids, which means that the very different DNA sequences can encode for the same protein. So you use that kind of flexibility in the genetic code to recode each of the proteins and then express them from promoters and other regulatory elements that are synthetic, that you have designed and know the exact expression levels of. And by doing this, you can then more easily modify expression of each of these proteins, and you don't have to worry about native regulation of the promoters that usually drive expression of these genes, or of things like small RNAs or methylation patterns that may modify the DNA itself or the sequences themselves. So this process of taking a bunch of native genes and recoding them synthetically has been used to do a bunch of kind of cool things in bacterial systems. So we've been able to engineer M13 phage, a specific type of bacteriophage, to function as an anti-tumor agent, to recode the entire gene cluster that makes type 3 secretion systems, which are what Salmonella bacteria use to inject effector proteins into mammalian cells. And by recoding the whole cluster under these synthetic controllable elements, these authors were able to move the ability to make this type 3 secretion system from Salmonella into other types of bacteria and to secrete non-pathogenic but other types of proteins through the needle. And there are several other examples in which refactoring has been really useful for allowing us to modify complex biological systems. So I think that we can use a refactoring type approach to try to engineer development. And this refactoring type approach is going to be based on auxin, or at least I think it would be cool if it was based on auxin. So auxin is a key plant hormone in development. It is synthesized and then moved around the plant in ways that change or dictate the structure of the resulting plant. So this is just examples of different portions of the plant, and the yellow arrows are showing sort of where auxin is flowing within those tissues, and then some of the proteins that are responding to that auxin concentration change. So in tissues or cells where there's low auxin, I'm going to present just a couple of the molecular tools that respond to auxin. So in cells that are low auxin, genes that drive development, some of them are kept off by the binding of auxin response factors, these ARF proteins, to DNA, and the binding of OXIA proteins to those ARF transcription factors. The OXIAs recruit repressors that keep the expression of the downstream genes off. And I should say, like, this is the canonical auxin response pathway. There are several variations on this pathway, but I'm just presenting to you the canonical one. So in tissues where there's high auxin, this OXIA protein becomes bound to this tier 1 E3 ligase, ubiquitin E3 ligase, which then leads to its degradation. The degradation of the OXIA frees up this ARF to recruit an RNA polymerase and initiate the transcription of downstream genes. So what is exciting is that we might be able to use mutant versions of these OXIAs and ARFs in order to control development. So if you mutate specific regions of these OXIAs, you can make them immune to tier 1 and the presence of auxin, so that even when there's a high amount of auxin in the plant, these proteins will continue to repress the ARFs that they usually bind to and prevent the transcription of downstream genes. And sort of similarly, but in an opposite direction, if you truncate off the region of the ARF that is bound by the OXIA, you would have a version of this protein that cannot be repressed, and therefore wherever it is expressed, it should initiate the transcription of its downstream genes. We're fortunate in that these mutations that would do each of these things have been well characterized for a large number of OXIAs and ARF transcription factors. So it should be relatively straightforward to figure out how to modify these proteins in order to make them insensitive to auxin and therefore useful for engineering development. Now, you have to be careful because messing with auxin response regulators can have a lot of pleiotropic effects. These are just two examples where you've got that kind of gain of function or auxin insensitive mutant version of an oxia that causes a bunch of developmental defects in this plant. So this is the plant containing the mutant version, and it is expressed from its native promoter, which is active in many cell types. And you can see that the plant doesn't produce any lateral roots, it has a hard time sensing gravity, doesn't have any root hairs, and has just a bunch of different pleiotropic effects. And this is an example of an ARF knockout where the root system fails to initiate, which is obviously a very large phenotype and not super useful for being able to modify specific aspects of development. So what I think you need to do is to carefully control the expression of mutant ARFs and oxias in order to make specific changes to the development of the plant. This is just a little cartoon suggesting that you would have this synthetic upstream gene expression program that eventually expresses an ARF or oxia that is insensitive to auxin. These are just two examples of how this may work. So in this example, you would be expressing the oxia mutant version in the xylem pole pericycle cells that would usually initiate lateral roots. But if you repress auxin signaling in those cell types, you should be able to make a plant that doesn't have any lateral roots, and restricting expression of the gene to just those cell types may enable you to have a very specific effect on development. So you can modify lateral root branching without modifying other aspects of the plant. Here's just a different example where you may affect gravitropism by expressing this same protein in a different tissue, in this case, in the epidermis and the elongation zone. So what's exciting, I think, about this strategy is that you wouldn't need to do a knockout. So in the refactoring pipeline that I showed you earlier, when people take a bunch of genes that encode for a complex biological process and then recode and reintroduce them, they do it into a knockout background. You have to remove all of the native genes that typically encode for this process. But here, because each of the oxia and ARF mutant proteins are dominant, you should be able to introduce this into a wild type background and modify development, which is nice because that means that the kind of plant material that you start with to do your engineering can be unmodified. Okay, so here's just an example of maybe a slightly more complex synthetic program. This is taking a background or the background plant that I showed you before where there's no lateral roots being produced, and showing how you might be able to reintroduce root branching under synthetic control using the kind of timed expression of the ARF proteins that are known to be important for lateral root development. I should point out again here, we really don't know if expressing these mutant ARF proteins even in the right cell types in the right order would produce lateral roots, or if there are more aspects of the development that we would need to be able to control. But it's just an example to show you how we might start to do these things. And my hope is that if you started to do this type of engineering, that it would elucidate potentially parts of development that we don't currently understand and don't know that we don't know. Okay, so why would we engineer auxin response to change plant development? There are a couple of reasons that I want to highlight that I have been sort of driving my interest in this space. The first is that auxin is sort of the key driver of development. Many of the environmental signals that alter a plant's development funnel down into auxin response regulators, whether that's modifying the expression of these regulators or their post-transcriptional modifications, all kinds of different things. And if we can engineer plant development to proceed independently of auxin, then we can engineer it to proceed independently of these other environmental cues. And so you get something maybe that looks like this, where the amount of water and nutrients, the presence of microbes, the growth of the shoot system may usually impact the growth of the root system. But if we genetically determine the root system so it always grows into the same shape, then we can start to probe structure-function relationships and say, how does the shape of the root system affect the plant's ability to survive in a specific environment, for example? And And then you get situations like this, where you, in a wild type plant, may have a root structure that grows differently in a bunch of different environments, but an engineered plant that always grows exactly the same root system can be used to see what the effect is on the shoot. Obviously, most of these plants will be very ill adapted or unable to survive in these environments, but I think it would be really interesting to use this type of engineering in order to test some of our hypotheses about what ideal root structures look like in different environments. Okay, the other reason to try to engineer plant development with auxin response regulators is that it may enable the engineering of diverse plant species. So we can come up with a scheme for Arabidopsis or for some of our model plants, where we show that the expression of mutant oxidase and ARFs can be used to modify development in sort of a predictive manner. Then we can look for oxidase and ARFs in other plant species, make the same type of modifications, and then drive modifications to their development as well. And this is useful because these genes are really highly conserved. And so unlike some of the developmental regulators that we have been able to modify in our model systems, which don't have homologs in these other species, oxidase and ARFs definitely do. In fact, genomic sequencing has revealed families of ARFs in all of the plant species or all of the flowering plant species that we've looked at to date. And this is where this sort of ties back into the plant cell atlas. So you'll notice that there are large families of ARFs and oxidase in each of these plant species, and it's really difficult to know which member of a family is important for the development of which structure in a plant. To a certain extent, they're interchangeable, but they're not completely identical. And so you can use some single cell omics to determine which oxidase and ARFs may be expressed in the tissue that you're interested in modifying, and its expression or modification in response to different conditions or over developmental time, which give you some idea of how it's acting and how to modify its expression in order to change development in a hopefully predictable manner. Okay, so that's my crazy scheme for how to modify plant development. Just in summary, the goal would be to engineer plant form by carefully controlling the expression of mutant auxin response regulators, and that you might want to use this to test structure function relationships or produce some new plant varieties. I'm just showing here a cartoon of one of the kind of original goals of original but very ambitious goals of synthetic biology, which is to engineer trees so that they grow into homes by modifying their growth program. So with that, I just want to say thank you. My lab just opened, so my thank you slide has just one person on it, but I also wanted to highlight Jose Dineni and his lab, where I recently finished a postdoc and got into plant biology. So that's it. Thank you.
Okay, great, Jen. That's some really interesting food for thought. I'll start here with a question. So you're focused on auxin, and clearly through that hormone you can control a lot. But we know that it's got such intricate feedback mechanisms, and so that works on both ends. On the refactoring side, figuring out the entire pathway in any given process is going to be, we don't have that information yet, right? And then when you're later on, when you're just manipulating a few, tweaking parts of the auxin pathway, knowing the consequences is going to be hard to predict. So how do you deal? It's kind of an open-ended question, but how do we deal with that in this approach?
Yeah, so one of the things that I like about these auxin response regulators is that they are so powerful and able to modify so many different aspects of development that they should have an impact at, at kind of wherever you express them. And the trick is to refine that impact down into the change in development that you want. And so we have some ideas based on like decades of auxin biology research, like which tissues are expressing which kind of auxin responsive transcription factors and things like that. So the idea is to start by testing some of the models of development that have been developed by expressing these things in a subset of tissues, and then looking at what the impact is by doing that on the whole plant. And if you see that there are more pleiotropic effects than you were expecting, then you can maybe try to modify the expression level even further by tuning not just the tissue in which it's expressed, but the point in development at which it's expressed. And quite honestly, I don't know that this would work, but I think it would be really fun to kind of put some of that knowledge to work and see, okay, this is our model of how the development proceeds. If we try to introduce an auxin response regulator to modify that development and we don't see what we were expecting, can we do some transcriptomics to see, okay, well, in normal progression of development, we know that auxin signaling activates this set of genes, but when we introduce our truncated ARF, for example, that is supposed to kind of mimic that auxin bolus that leads to gene expression, we see only a subset of those genes expressed, or we see, all the genes expressed that we were expecting, plus a whole bunch more. It gives us an opportunity to start to try to maybe troubleshoot and refine and broaden maybe our understanding of how these processes are working.
Cool. So it's kind of also an experimental platform to learn more about the plants. Okay, let me move on to some other questions. Here's an interesting one. Basically, I'll boil this down, is you sort of propose making either more simplified or replicating circuits. Do you ever want to make them more complex?
Yeah, I think if you could get the simple things to it. So yeah, one of the reasons I focus on the simple stuff is because even the simple things are very hard. But it would be great to see what would happen if you could modify sort of a simple circuit to kind of include more environmental inputs or more decision-making points that then give you, not just a modified version of the original plant, but a completely new form. I don't have the best idea at the moment of what that looks like. I think the challenge of trying to just recapitulate normal development using synthetic regulation is going to be challenging enough, and that we would learn a lot by modifying our expression programs to change dynamics of these pathways or tissues in which they're expressed, which I think still constitute relatively simple circuits based by this kind of metric, but would be pretty informative. So maybe if we get there, would love to see more complex stuff happen. Be awesome.
Okay, cool. All right, here's kind of a related idea or thought, and that has to do with how do you deal or factor in or break apart the crosstalk with other hormones and auxin?
Yeah, so the refactoring process of recoding the genes is one way in which I was hoping to try to break the interaction between auxin and other hormones. So we know that other hormones can lead to the expression of small RNAs that regulate auxin response factor and OXIA levels, and also that they can lead to post-translational modifications. So if you change, residues within these mutant OXIAs and ARFs that are known to be phosphorylated, sumolated, all these other things, then you should start to wall off these regulators from most of the other inputs that would change their activity. There's going to be a limit to this, but it's a place to start and may also help us figure out, other mechanisms by which these other hormones are modifying development through ARFs and OXIAs.
Cool. Again, like another experimental, using it as an experimental system, a probing system. Cool. Okay. We have more questions, but we're over time now, and I guess we promised to let everybody go. And I'm not sure if the speakers can keep answering the questions in the Q&A, but maybe they can, and so maybe they will. And I think Mary wants to close off with a couple of comments.
Yes, thank you. And I can send the questions in chat to the speakers if they would like to continue answering them. First of all, I'd like to say thank you to Hilda, Daniel, and Jen. Great talks. And thank you, Kenneth, for fielding the questions expertly. Thank you, attendees. There was a lot of chat, a lot of questions. It was very, very stimulating. The recording will be posted in the next few days. And this is really one that I think we want to all encourage our students to watch because, you know, there's no plants are boring going on here, right? This is exciting stuff. This is the future. I'm really excited. And so that's it. Thank you, editors. Thank you, Plant Phys. Thank you, ASPD. Thank you, everyone. And I'll see you next time. Bye-bye. Thank you.
Great. Thank you.
Thank you.
Bye.
Bye-bye.
Thanks a lot.